In this episode, I explore the process of protein synthesis. This process uses DNA to create proteins by reading the genes. These genes are translated into proteins. Remember, proteins are made of polypeptide chains, and this process can also be referred to as polypeptide synthesis. Now let's look at some key terms that will help you understand the concepts presented in this episode. Recall that a nucleotide is a unit that makes up DNA and, in some cases, RNA that consists of adenine, thymine, guanine, cytosine, which is a base, phosphoric acid, and a sugar molecule. These nucleotides code for triplets. They are a sequence of 3 that code up for a codon in transcription that are then translated in protein synthesis. A gene is a section of the chromosome that codes for a protein and that has within it transcriptional units. This section has introns and exons, which I'll explain later in the episode, which code for protein production. Polypeptide chains are chains of peptides that make up a protein. In the image shown, it shows how a peptide becomes a functional protein by being folded into a more complex structure, which allows for the protein to carry out its functions. I'll explain this in more detail in a future episode on the structure and function of proteins. So let's sum up some concepts from this introduction. DNA triplets code for mRNA codons, and genes code for mRNA transcriptional units to then produce a functional protein. Now let's look at transcription and translation in more detail. Transcription is the process where the code contained in the DNA molecule is transcribed into an mRNA molecule. Remember that the biological process like DNA replication and protein synthesis are under the control of metabolic processes. These metabolic processes are actioned by enzymes. In this case, RNA polymerase is the enzyme that makes up the mRNA strand from DNA. Some key features that's helpful to understand: only one gene length is transcribed at a time. There's a start signal and a stop signal at each end. Only RNA polymerase is involved in mRNA synthesis and in the unwinding and rewinding of DNA. mRNA synthesis happens rapidly due to many RNA polymerase activating along the same gene section at the same time. There are 2 main steps in transcription. There's pre-messenger RNA that's formed and the splicing and editing of pre-messenger RNA to form a mature mRNA molecule. Like DNA replication, transcription involves the unwinding, then copying. However, there's only one strand being created, and that's the copy of the sense strand. The sense strand makes sense when it's later translated into a protein, and we'll look at this later in the episode. Due to the sense strand being replicated, it's actually the antisense strand that's being transcribed. Remember that because of the complementary nature of bases, protein synthesis will occur along the antisense strand when it's transcribed. For protein synthesis, the pairing of bases is similar to that of DNA replication. Guanine pairs with cytosine, however, adenine is actually paired with uracil rather than thymine. Ribonucleoside triphosphates, NTPs, align along the antisense strand of DNA according to their pairing. RNA polymerase joins with ribonucleotides together. At the stop signal, the replication stops and the DNA rewinds. This process forms the pre-messenger RNA, which contains both introns and exons. Introns don't code for proteins, and exons do code for proteins. So the pre-messenger RNA is then spliced together to only contain the sections that code for proteins, producing a mature mRNA strand. mRNA formed by transcription is transported out of the nucleus into the cytoplasm. Ribosomes use mRNA to direct protein synthesis via the process known as translation. You need to recall that RNA nucleotide triplets are transcribed into codons within the mRNA that then get translated into anticodons that then form polypeptide chains. An example would be a triplet of TAC forms a codon of AUG that then forms a code for an anticodon of UAC that then codes for a peptide. Now let's look at translation in more detail. As mRNA passes through the ribosome, each codon interacts with an anticodon with a specific tRNA. tRNA, known as transfer RNA, is a cloverleaf-shaped structure that is specific to each amino acid. The amino acid attaches to the 3' OH group of the tRNA. Due to the structural nature of tRNA, the enzyme aminoacetyl tRNA synthase adds the correct anticodon to the tRNA to the mRNA, which means that this forms the correct polypeptide chain, which then becomes a protein. From this clip, you'll be able to define polypeptide synthesis, describe the role of peptide synthesis, describe the steps of transcription, translation, and suggest reasons why the process of polypeptide synthesis is important. Now think, if an error occurred in DNA replication or protein synthesis, for example the wrong nucleotide being matched, how would this affect the organism and further protein synthesis? Think in a bigger picture, why is polypeptide synthesis important? See you next time, budding biologist.